Hey everybody. Hey, it's Friday. Woohoo. And we are nearly done shaving our metaphorical yak. So if you don't know what that means, look up yak shaving on the internet. It's a programming thing, apparently. Anyway, so we have got ourselves to a point where I think we've done a pretty decent analysis of this RNA-Seq data. So we are at the end of our 8th week, and over those 8 weeks, we have installed a virtual machine, learnt to use a large number of very commonly used command line tools, learnt how to use the command line to download data, looked at excuse me, sorry. Looked at downloading data from UniProt, doing multiple sequence alignments, then downloaded data from the sequence read archive, looked at QC of that data. We've then quantified it using Salmon. Then we've done a fairly decent introduction to R, basic introduction to basic R. And then we've used R to read that count data in and analyze it with DSeq2 to give us differentially expressed genes, which we have now started to visualize in various ways. And in 8 weeks and around about 36 hours or so of lessons, I think that's really pretty good progress. So yeah, we've covered an awful lot of ground in those 8 weeks. And it is, I do now need to seriously think about what we're going to do next week, because I think we're getting towards the end of this kind of introduction to RNA sequencing analysis. We've kind of done RNA sequencing analysis. I think it would be useful and instructive to analyze the second RNA-Seq dataset that we had that we've downloaded. At the moment we've only analysed these first 6 NHBE samples, and I think it would be good to analyse the other samples as well, and maybe to use those samples, use that analysis and compare between the analysis that we've done this week and the analysis of that second cell line. I think that would be really helpful. And interesting to do, but then we do really need to think about what we're going to do next. So I've had lots of suggestions. The survey that I started last week has had, I think, getting on towards 100 responses now, and lots of people have requested lots of different things. And so I'll take a look at what people have asked for and think a little bit about my own expertise as well, and where I think I think we can go next. And in the next couple of weeks, we'll start to think about that next process. It might be that we'll take maybe a week or so to do some kind of some more fundamental technologies, so to talk about Git and GitHub and maybe R Markdown and things like that we haven't really had a chance to cover yet. I think that would be quite useful to do before moving on to any significant new kind of type of analysis, any new biological technology. But yes, I think if you've got this far and you want to self-identify as a bioinformatician, at least a kind of a dabbler in bioinformatics, then I think that's perfectly reasonable. Okay, so with all of that said, what is our plan for today? What are we going to do to round off this week? And what I want to do is turn our attention a little bit to the function of these genes that we've identified as being differentially expressed. So at the moment, my disclaimer here is I am a bioinformatician. If you remember right back at the start, I think I talked about my training, and I am a geneticist by training, and I studied Drosophila development in my PhD, and the genetics of Drosophila development. but that was a long time ago, and I've been a core facility bioinformatician for pretty much the last 15 years. My expertise these days is in bioinformatics, not really in any specific area of biology, especially as the analysis that I do is very wide ranging and comes from a lot of different systems. So I don't I I think of my biological expertise these days as kind of broad and shallow rather than focused and very very deep. So when I'm confronted with a list of differentially expressed genes, I can't immediately identify the interesting genes in that list and the ones that are connected with the biology that we're studying. Although I don't necessarily think of that as a handicap, because I think there can be a little bit of a tendency when you're very intimately familiar with a particular area to cherry pick a list of differentially expressed genes and sort of decide a priori which ones are interesting on the basis of your prior knowledge. So coming to a list of differentially expressed genes without that prior knowledge, which I suspect a lot of us are doing with this particular experiment, can be quite useful because it gives you this kind of unbiased view of the world. But then you do need some way of taking that list of differentially expressed genes and inferring something from it about the functioning of the system that you're analysing. And the way that we normally think about doing this is some kind of functional enrichment testing. So there are lots and lots of ways of doing enrichment tests, and we're going to look at one particular way this afternoon, and we're going to look at certainly one set of databases up front. So what we're going to do over the next hour or so, 50 minutes or so, is have a look at the Gene Ontology and we're going to look for enrichment of gene ontology classification in our differentially expressed genes. There are lots of ways of doing this. We're going to use one particular statistical test called the hypergeometric test to do it, which is a common way of doing it. This hypergeometric test is sometimes also called the Fisher's exact test. They're interchangeable under most circumstances as statistical tests, and that's how we're going to do it. There are all sorts of really fancy commercial packages for doing this kind of analysis. In our unit, we have a license for QIAGEN's Ingenuity pathway analysis, which is a fancy, expensive commercial way of doing very similar things. It has some kind of fancy add-on features that make it worthwhile paying the exorbitant license fee, but nevertheless the basic ideas behind it in terms of functional enrichment are the same as we're going to be applying this afternoon. So I'm going to introduce a few concepts, and then we will talk about how we're going to apply this in R. So first of all, let's have a look at the Gene Ontology. So the Gene Ontology is the idea behind the Gene Ontology is that it's a comprehensive dictionary of terms relating to biological systems. And what that means is you can attach, the Gene Ontology comes in three different sort of arms. There's three different components to the Gene Ontology. This homepage is not particularly great for describing it, I will admit. So there's the biological process Gene Ontology, sometimes referred to as BP, the molecular function Gene Ontology, and the cellular component Gene Ontology. So these three different sort of arms of the whole thing they're independent from one another. They're separate control vocabularies, but they describe obviously these different facets of the function that gene products have. So the biological processes that they take part in. So that can sometimes be thought of as kind of things like pathways and so on. The molecular functions that they have, so that's the things that the gene products themselves do. So are they kinases or phosphatases or proteases or whatever else, whatever other functions gene products can have. And where do you find them in the cell? So where are they? What's their subcellular location? So do you find them in the nucleus or in the cytoplasm or in the extracellular matrix or whatever other, again, whatever other locations you might have. And so what we're dealing with at this point is really lists of things. So we have a list of differentially expressed genes that we've identified via our what's the word that I'm looking for? Statistical tests that we've applied. So DSeq has enabled us to identify differentially expressed genes in the system that we've tested. We're now kind of adding to that a second list, and that is a list of the functions that those genes undertake. Thanks, Pia. Which is actually not very informative. And I was just trying to. There we go. There's my whiteboard. Right. Okay. So. Right, so we've got a list of genes. So these are our ensemble gene IDs that we have a list of. And what we're going to do this afternoon is we're going to attach those genes to a list of functions. So these will be Gene Ontology IDs, which always begin with a GO and then a colon. And so we're going to associate between these two lists, a list of genes and a list of functions. One gene may have many functions, so one gene may map to many gene ontology terms, but essentially we're going to try and draw links between these lists, and then we're going to use those links to try and understand whether any of these functions are enriched in our differentially expressed genes. So whether we're identifying that function more often than we would expect if we had picked our differentially expressed genes purely at random from our genome. And to do that, we're going to be using a statistical test called the hypergeometric test. Hypergeometric test, or Fisher's exact test. Okay, so this is the stats that let us work out whether we're looking at statistical enrichment of a particular process. The way that this statistical test is usually visualized is as a pot of balls. Okay, so I'm going to draw a pot of balls. So you've got a pot which contains a set of balls. What we're going to do is we're going to do an experiment that draws balls out of our pot. Okay, so we're going to extract balls out of our pot. Okay, YouTube is telling me my live stream is offline. Just let me know if you can still hear me. And I'll wait a few seconds just to see if everybody can still hear me. Because YouTube's being funny again. Meanwhile, I will continue to draw my picture because these balls have different colours. That's the key to the whole thing here. Colour these in. Everybody still hear me? You still there? I can see Peter saying hello. Excellent. Right, good. Sorry, I didn't want to be waffling away and for you not to hear me. That would be a tragedy, a Friday afternoon tragedy. Okay, so I nearly finished colouring in my balls anyway. So, right, what we're going to do is we're going to have a process which pulls balls out of this urn. Now, in the situation that we're dealing with here, in the experiment that we're dealing with here, the balls in the urn, so the red and the black balls that are in our urn, in our experiment, these are genes. Okay? So the balls in our pot, they're genes. The process which pulls the balls out of the pot, that is our that's our differential expression test. So we've got all of our genes in our pot, and then we test for differential expression using DSeq, and that pulls balls out of the urn. So the balls that come out of the urn are the balls that are differentially expressed. And then the colours of the balls are either so the black balls are just just a set of genes. The red balls are the genes which belong to the Gene Ontology term that we're particularly interested in this test. So the red balls in this case are the viral response genes, for instance, to pick a Gene Ontology term out of thin air, and we want to test whether viral response genes are enriched in the genes which are differentially expressed. And so we do our differential expression test, and that pulls a set of balls out of our urn. So let's say we pull 8 balls out. Okay, and obviously the balls that come out of the urn will have different colours. So we might pick. So we might have a black one and another black one, and then a red one, and a red one, and a red one, and a black one, and then 2 more red ones. Okay, so that's our draw. That is the balls that have come out of our urn or out of our pot when we have done our differential expression test. So those are our differentially expressed genes. And of our 8 differentially expressed genes, so we've got 8 differentially expressed genes. 3 of them don't contain, aren't annotated with our function of interest, and 5 of them are annotated with our function of interest. And the question that you ask with the hypergeometric distribution is, given this distribution of balls in our draw, so 5 out of 8 being red, what are the chances of that draw being the product of a random process? Okay, so if we look at the population of our whole, of our 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29, 30, 31, 32, 33, 34. 34. There are 34 balls in the urn, 7 of them are red. So, given this population of balls, if we draw 8 balls out of the urn without replacement, that's key thing here to this statistical test. We draw 8 balls out without replacement. What's the probability that in that draw we get 5 red balls and 3 black balls? And in terms of this particular distribution of balls, where 7 of 34 are red, drawing 5 out of 8 as red is quite a low probability event. The chances of that happening, if this was a random draw, are very low. So what you'll find with the hypergeometric test in this situation is that this isn't a random draw. That we have selected when we've picked these 8 genes, we have enriched for the genes carrying the function that's denoted by the red color, which I said before was viral response. So Graham said, isn't this a binomial problem? It's very related to the binomial distribution. So the binomial describes the probability of a series of successes in draws from a population with replacement. So you take the ball out, you see if it's the colour that you want, you register whether it's a success or a failure, and then you put the ball back in to the pot. But the hypergeometric is without replacement. So you take the ball out and you keep it out. And that's the big difference between the binomial and the hypergeometric. So they are extremely related statistical distributions and statistical tests. So in the case of the hypergeometric, we're taking the balls out of the urn when we draw them, and not putting them back in. So I'm just gonna rub them out to be there you go, 3, 4, 5. So there you go. That's, you got to imagine that we've taken the genes out, and that's because when we test, gene can be differentially expressed or not differentially expressed, but we're not considering it more than once in our statistics. So all of this is without replacement. Okay, so that's the hypergeometric test, and we're going to be using the annotation for the gene ontology with our knowledge of this hypergeometric test to find out whether we have any statistically enriched gene ontology terms amongst our differentially expressed genes. And my kind of usual disclaimer with all of this stuff applies, which is I haven't tested this yet. I've made sure that I know exactly how I'm going to do the processes that we're going to do this afternoon, but I haven't done them before with this data set. So I don't know the answer to the question that we're going to be asking. I don't know whether we're going to find any enriched functions or not, and what those, if we do find them, what those enriched functions are going to be. So we're going to explore this data set together. So as with all of these functionalities in R, we're going to need a package. And there's actually quite a lot of packages that do this, that apply these kind of statistics in Bioconductor. Historically, I've recommended a package called GoStats, which is quite a nice package for this analysis, but quite complicated. What I've used now and what we recommend now is a package called clusterProfiler, mainly because it applies these statistics in the same way as GoStats does, but it's a little bit more flexible and lets us test more things other than just the gene ontology, and it's also some okay visualizations built in package, and it's clusterProfiler with a lowercase c and an uppercase p. I'm afraid the rules of how capitalization works in how the capitalization works in the name of R packages is extremely random. Different developers have different ideas as to how capitalization should work. So yeah, clusterProfiler with a capital P, all lowercase otherwise. And this has to install quite a lot of dependencies, so it's going to take a little while to install. The other thing that we need is an annotation database. So having spent yesterday, was it yesterday, day before yesterday, retrieving annotation data from Biomart, clusterProfiler gets its annotation data in a different way, which is slightly irksome. But we're going to have to install an annotation database into our R session as well. The annotation database that we're going to install is an organism annotation base that's distributed as part of Bioconductor. So we're going to grab that in just a second, just waiting for clusterProfiler to install. So yes, this enrichment test you absolutely can do on the Gene Ontology website as is suggested. You could paste your 141 ensemble gene IDs in here and run. It's called Panther, is the Gene Ontology's own tool for doing this, and get perfectly decent results. Actually, probably quite good results for this kind of process. The reason for doing it in R is partly just staying within the same environment and being able to take your differential expression results straight from your test into the next package, and to be able to script that as well. But also the visualizations, some of the visualizations that are available to us, which can be quite useful and quite handy to be able to generate those directly in R as well. Okay, so that's installed clusterProfiler. So first things first, let's load that package. So always have to remember to attach the package once we've installed it. So clusterProfiler up there. We'll load that, and then we need our annotation database. So as I said, we're going to install an annotation database that is part of Bioconductor. It's a Bioconductor package, and it is called, so let's do Bioc Manager install. It's called, this is a complicated one, org for organism, .hs with a capital H for Homo sapiens, it's the human database, eg for Entrez gene, and .db, and it's got lots of dots in. So org.hs.eg.db, and that's this is an annotation package. So the DB stands for database. So it's an annotation package from Bioconductor that it's going to download and install. These annotation packages can be quite large. You can see this one's nearly 80 meg, so it's a decent sized package. I saw a thing on Twitter today which was a debugging guide, and there's sort of 3 things that you can do to debug your code under most circumstances. The 3 things that you can check are: 1, is it a typo? 2, is it to do with your parentheses? And 3, is it to do with your capitalization? I think was the third thing. So yeah, those things are always the things that catch you out, I think. So that annotation package is downloading. And we're nearly there. So then we want to attach that as well in our package loading, because we want to make sure that that's available to us. Longer installations today for some reason. Lots of dependencies and one quite large package. So that's done, and we'll attach that one as well. Now this is an interesting thing. So I'm going to draw your attention to this message that popped up when I loaded this package. Okay, so library org.hs.eg.db. Okay, when I loaded this, it has attached a required package, which is annotation DBI, and annotation DBI has masked objects from packages that we've already got attached. So what it says is the following object is masked from package plotly, and that's select, and the following object is masked from package dplyr, and again that's select. And what this means is that these 3 packages, so plotly, dplyr, and annotation DBI, all have a function in them called select. And when you try to use the function select in your R session, R will pick the one from the package which you attached most recently. So when you load a new package, any functions which have the same name in that package as in other packages which you've already attached or in your base packages, the packages in the functions in those other packages will be masked, which means they won't be the first function that R uses when you try to use. So when we now when we try to use the function select, R will use the version of select from the annotation DBI package, not from the dplyr package or from the plotly package. And all of those select functions will do different things on different types of data. So if we ever now want to use the dplyr select function, we have to be explicit about it. We have to say that we're using select from dplyr by doing this 2 colon arrangement. So the name of the package, then 2 colons, and then the name of the function that we want to use. dplyr in particular suffers from this problem quite a lot, which is that quite often they use commonly used function names like select and filter and various other things as well. And quite often it will be the dplyr one that you mean. But if you've loaded another package after dplyr, like we have here, you won't be using the right function. And actually, somebody emailed me about a problem this afternoon that I think arose exactly from this problem, that they were trying to use select, and I suspect they'd loaded plotly after they'd loaded dplyr, and so they weren't addressing the correct select method. So I have got into the habit recently of when I use functions from dplyr, of using this explicit version of calling the dplyr functions. The one way that you can be strict with yourself about it is to always load the tidyverse last. So have the tidyverse at the end of your package loading, so that dplyr gets loaded at the end. So at the moment it's up here on line 5. But obviously when you're developing a script like this and you're doing things you're not running the whole script all at once, but you're kind of developing it iteratively, then it can be quite difficult to remember to make sure that your package loads all happen in the right order. So sometimes it can be good to get into the habit of calling functions explicitly like this, especially when they have common names like select and filter and various other things. Anyway, that's a little sidetrack. I didn't want to get into too much, but okay. So clusterProfiler. So we've loaded clusterProfiler, and we're going to use it to test for Go enrichment. So clusterProfiler has a function for Go enrichment, which is imaginatively called enrichGO. Okay. So this is the enrichGO help here, and describes to us a load of arguments which we might want to change or make use of. So Gene, first of all, it says a vector of entres gene IDs, but actually key type allows you to change what type of ID it is that you use. I think if we come on to look at other databases as well, having the entres gene IDs may well be useful. So it may make sense for us to retrieve these from Biomart. So I think we'll start by doing that. So we'll retrieve the entres gene identifiers from Biomart. We could give the key type here, this key type argument, we could set to ensemble and use our ensemble gene IDs. But that doesn't work for every type of database that you want to test against, whereas the entres gene IDs almost always do. So we're going to retrieve those from Biomart, actually, as it goes, I think. We need to give it our orgdb. That's the database that we just attached, the org.hs.eg.db package that we just attached. We need to say which ontology we want to test. So by default, it's molecular function. I think we'll probably look at the biological process ontology first, and some various other things as well. So let's go about running this. So enrichGO. Let's call it ego. Actually, that's the thing that they usually use in their documentation. Okay, here we go. The function is enrichGO. We want gene equals org db equals ont equals, then we want universe. That's the other thing that we want. I'll explain that in a second. And let's set actually, let's leave everything else as the default for now and see how we get on. Okay, so that's my set of arguments that I need to define. So the first thing we need is a set of interest gene identifiers for our differentially expressed genes. So let's go back to our bioconductor and we'll figure out how to retrieve these. So bioconductor was a couple of days ago. Here we go. And get BM was our call, if you remember. So let's copy this and paste it down here. Right. Okay. So let's call it entgene. We don't need any of these things. We don't actually need any of these things. We've already retrieved all of this annotation. We need the entres or entree, depending on how you like to pronounce it, gene ID. I've actually got the list attributes still open here. So that's the attribute that we need. And there it is, number 81, okay, entres gene ID. That's the attribute that we want to retrieve from Biomart. So let's put that in there. We're going to filter on the ensemble gene ID like we have done before. And the values that we're going to pass it are going to be our the values are going to be our differentially expressed genes. So that is in which one was it? It was anno_df3. df3, the ends gene column. Okay, that will look up those 141 identifiers and retrieve the interest gene ID back for them. We don't need to worry too much about retrieving the ensemble gene IDs alongside these. It doesn't really matter for this purpose. So we're just going to get the interest gene IDs. So we'll run that, and that will run quite quickly because there's only 141 of them. And so you'll see that one of our ensemble gene IDs is going to map to an interest gene ID. And there's good reasons for that. The interest gene IDs are not as complete, and especially for things that are relatively unannotated, there often isn't one. And so I'm not surprised that there's only 125 of them, and it's not really too much of a problem. I expect to see some loss of data when we're running this. Okay, so that's as a data frame with one column, and we need it as a vector. So let's just pull that vector that column out as a vector. So ent gene equals ent gene dollars interest gene ID. Okay, so that will give me the just a vector of those gene IDs. There you go. So that is my gene argument. The genes that are differentially expressed, the gene list that I want to test. That is the balls that have been pulled out of the pot during our expression, during our differential expression test. But the other thing that that enrichGO wants to know is what was in the pot in the first place. So you could assume that the pot is just all of the genes in the genome, but actually that's not entirely reasonable for the test that we've done. It makes more sense that actually what's in the pot is the genes which we've actually tested for differential expression, and that is our 16,000 or so genes in ANODF1. So we're going to do the same thing here as we did here, and we're going to call it ent-uni for the universe. But instead of DF3, we're going to do it for DF1. So this is going to pull back all of the Entres gene identifiers for everything that we tested for differential expression. So everything that passed all of our filters before we tested for differential expression. Why is ANODF1 not found? Is that because I didn't call it? I called it annotated DF. Because, you know, why be consistent when you can be inconsistent? So that's going to take slightly longer because it's a bigger query. But the results are going to be essentially the same. It's going to be a data frame with one column that's going to consist of Entres gene IDs. And again, there will be fewer of those than there were input ensemble gene IDs. But again, that still isn't a significant problem in the context of this particular test. So n uni dollars Entres gene ID to give us our universe. So this is our universe. So all of the genes which we tested for differential expression. OrgDB is the package that contains the annotation. So this is the thing that maps those Entres gene identifiers to the Gene Ontology identifiers that we want. So that's our second list. That's our list of annotation is contained within that database. And then the ontology that we want to test, we're going to test the biological process ontology. There's no underscore between Entres and gene. It's just Entres gene ID with no underscore. Just referring to Graham's question there in the comments. So yes, that's all of our arguments for our enrichGO function. So now we're going to run that, and this has got a decent amount of computation to do. So this will take a couple of minutes to run because it's got to do an awful lot of testing. So it's retrieving it's putting the genes into their sets by their gene ontology annotation, and then for every gene term in the biological process gene ontology, it's testing whether that term is enriched or not. It's got quite a lot of work to do to figure out what's what. Biological process terms in the biological process gene ontology. Universe is not in character and will be ignored. Or does that need to be a character vector and not a... Hold on. I didn't particularly want to see that as character and uni. Okay, so it's not a character? Oh, okay. What is it then? Is it a double? I know they're numerical. I know it's not a double. Factor? What the hell is it then? Integer. Great. Okay, so we need to cast that to a character. Let's do it with both of them. As.character. See how it treats us there. As.character. Yes. Okay. Oh yeah. And gene equals as character. So it's retrieved the type of data. It's retrieved them as integers, which is not unreasonable. They're all numeric and they're all whole numbers, so it's not completely unreasonable that they're integers. But I think that enrichGO is expecting them to be characters, so we need to make sure that they're set as the right type, and so as.character will fix that for us, I hope. Try that again, and again this is going to take a couple of minutes to run. So this will produce the output of this. Is, I think it's quite a complicated output, but it's able to be summarized as a table. So we will do exactly that once this is run. Okay, I didn't get the warning this time, which is good. So let's have a look at our results. I'm going to make to probe the results a bit, I'm just going to make the console bigger because I think the results might be, if there are results, I think they might be tricky to look at in a little console. So there we go. We do have some results. It's an overrepresentation test. There's some data in here. So the organism, the ontology that we've tested, the key type that we had, genes in there. These are our Gene Ontology IDs, and these are descriptions, and there's some various other things in there. I think if you do a summary of that object, do you get the table itself back? Yes, you do. So view that to have a look at the table of these are the enrichGO terms in our test. So okay, so quickly go through the columns in this and roughly explain what's going on. So this is the Gene Ontology ID that is the subject of the test in this row. So each row is a test. This is the description of that gene ontology term, so response to bacterium. Okay. Then we've got the gene ratio. So this is how many genes in our gene list are annotated with this term. So in our 117 genes that we've ended up giving to enrichGO, which is lower than 141 because there's a little bit of so of those 117 genes in our selected list, so in the balls that we've pulled out of the pot, 28 of them are red balls. So 28 of them have this annotation response to bacterium. In our universe, so the background ratio, there are 368 genes in total out of nearly 12,000 genes in our universe, which carry this annotation. So you can figure out the ratios of the number that there are in our set versus the number that there are in our universe, and then what we're seeing is an enrichment over that of genes in our gene list over genes in the universe. The p-value for that is then derived from the hypergeometric test. Okay, so that's what this p-value is derived from. And then we've got a couple of different multiple testing corrections applied. The q value is the one that we're interested in. That's the result of the Benjamin-Hochberg multiple testing correction. So again, this has been corrected for multiple tests, and you can see that there's quite a lot of results in here. There's quite a few gene ontology terms which have passed this particular test, even though quite a few of them only have a very small number of genes represented in their lists. So this is quite a nice way of associating the genes in our gene list with some with some functional terms, some of which look like they might be meaningful in terms of what's going on. It's a response to a virus. Okay, we're getting a response to bacteria here, but there's other kind of inflammatory pathways and things like that cropping up in this particular set, which is interesting. So the other thing, show all panes, that I wanted to show you with this package then was the mechanisms it gives you for visualizing these results. So as well as being able to look at that table that results from this test and just looking through those as a table, we also get some visualizations that come in the clusterProfiler package. So for instance, there's one called bar plot, which you just give your results to. So this variable that we've just created called ego, we give that to our bar plot, and it will produce a visualization of the top, looks like 8 in this case for some reason. 2, 3, 4, 5, 6, 7, 8. Yes, 8 different gene ontology terms with I think it's the size on the x-axis. So that's how many genes there are in that category. Although I thought it was 17 before. That looks like more than 17. Anyway, and they're coloured in by the p-value. So you can change the parameters of this to configure the plot that it produces as well. So let's have a look at the help for bar plot. Let's see what we can change. We can change. Pretty sure you can change the number of things. None of this looks very easy. Is this the base bar plot? That's the base bar plot. We want clusterProfiler bar plot. Ooh, interesting. How do you do it? I'm going to have to try and remember now because it's not obvious from that documentation. It's no, I can't remember. I'm going to have to look up the documentation. Okay, bear with me for 1 second. Here, which is going to open in my browser, and then we got to go to the visualizations. Sorry, I'll put it on the screen. The visualizations here, we can jump to the bar plot. There you go, show category is the answer. That's the argument that I need to include to look at more. Sorry, that was a bit of a faff. There we go. So show category. So we can show more than just the ones it decides to show for us. And so get a slightly more in-depth view of the categories in that bar plot. Similarly, there's a plot called dot plot. Similar sort of idea, but shows you the count as well as the ratio and the adjusted p-value. That's quite a useful view as well. And again, show category will expand the number of gene ontology categories that are shown in that particular chart. These are ranked by some mechanism that I'm not immediately sure of. So the more you pull out, the more the further down that ranking it's going to go. Again, these are ggplot plots. So you can edit these by adding geoms and themes and things to them if you so desire, because they're generated using ggplot. The other one which a lot of people like is this CNET plot, connectivity net plot, which we will try and produce. This links. This doesn't work, apparently. Interesting. Why doesn't that work? Try it with a small number of categories. No, that's not going to work. Okay. It can be quite useful because it can show you the membership of the different the different categories by linking up the categories with the genes that belong to them, and you can see genes that belong to multiple categories and things like that. It can be quite a useful visualization. I'm not sure off the top of my head why it isn't working, and I will try and fix that and come back to that next week, I guess, because we're fast running out of time. But that can be quite a useful plot as well. Okay. Right, so that's sort of visualizations there, and those can be quite useful. And this I should say this is kind of the user manual of this clusterProfiler package, and it's extremely extensive. It's very large and can be very useful. So I think it can be, it can be a really nice sort of visual package for both working with and visualizing these kind of enrichment results. We can also use it to look at pathways in KEGG as well, as you can see from some of this documentation. Let's just try this Go plot just as a last throw of the dice. No, that's not working for me either. It looks like it might be working for some of you in the comments, so maybe it's a problem that's particular to me for some reason that I can't quite figure out, because KEGG can be quite useful. So the equivalent for KEGG is not enrichGO, but enrichKEGG. So enrichKEGG. The idea is basically the same, but this time we don't necessarily have to point it at a database because it uses uses KEGDB, I think, which is already downloaded. This only really works with Entrust gene IDs, which is why I wanted to get those IDs at the beginning. So let's do an enrichment of the KEGG by doing equals enrichKEGG, where gene equals the same thing, so we don't need to change that. Organism is the default HSA. Key type, I'm pretty sure we can leave as the default as well. We'll stick in our universe and uni, and I think that's probably all we need, he said confidently. And this should hopefully give us enriched KEGG pathways as well. So let's have a look at the summary of that. Yeah, that looks like we've got something. There we go. So again, we're seeing inflammatory stuff going on here, really. So IL-17, TNF, I mean, these are fairly well-trodden inflammatory pathways. So I guess not massively surprising, but still can be useful to see. Okay, so that's KEGG pathway enrichment as well. And again, there will be methods um, for looking at seeing these pathways as well, and we can maybe explore that next week. Maybe I'll try and fix these visualizations, and we'll come back to that on Monday and do more visualizations. Okay. So that brings us to the end of week 8. And also really kind of we have, I think we'll retread some of this ground just to reinforce some of the messages from the last couple of weeks, because I'm aware that the pace has sped up slightly over the last couple of weeks, and so it might be nice to go back over things, but in the other data set to make sure that everyone is still keeping up. So I think maybe that's what we'll spend next week doing. But that we have now worked through the kind of all of the basic steps of an RNA-Seq analysis. If you were to get hold of your own RNA-Seq data or want to download some publicly available RNA-Seq data, essentially those are the steps that you will take. The QC, quantification, and then analysis in R that we've done over the last couple of weeks is the way that you would analyse that data. So it's nice to get to that point, and it's nice to get to that point on Friday afternoon as well. So with that, I will sign off. I wish you all a lovely weekend, and I will see you back here next week, I hope, for more of the same. And have a lovely time this weekend. Keep safe, stay well, and see you next week. Bye, everybody.